Welcome, everybody, to Eye on Education. I am Ben Shaberman, Senior Director of Scientific Outreach at the Foundation Fighting Blindness, and I'm pleased to present an introduction to stem cells. It'll be a pretty brief and basic overview of stem cells and how they might be applied to retinal degenerative diseases as a potential therapeutic approach. So let's get started. And I'd like to kick things off with just a basic definition of a stem cell. And often when we're thinking about stem cells, we're thinking about the pluripotent variety, because those are the most powerful and flexible stem cell type. And you can think of pluripotent stem cells as being like a clean slate, in that you can make virtually any cell type in the body that you need. Bone, muscle, skin, and obviously in our world of retinal diseases, we're interested in making new retinal cells, often photoreceptors or retinal pigment epithelial cells. And we'll talk more about that in the upcoming slides. So another advantage of pluripotent stem cells is that you can take a small sample of these stem cells and literally make billions and billions of the cell type that you need for potentially thousands and thousands of treatments. Now, pluripotent stem cells can come from different sources. There are the embryonic variety and a newer approach that's gained a lot of momentum, both in the lab and now in clinical trials, are induced pluripotent stem cells. And let's look at those on the next slide. So induced pluripotent stem cells are derived from a small sample of mature blood or skin taken from an adult. So again, mature blood or skin. And what researchers do is they genetically tweak these cells so they revert back to a stem cell-like state. So it's like turning the clock back on these blood or skin cells so they can become stem cells and then coaxed forward to become the desired cell type. In our world, again, we're most interested in photoreceptors or RPE cells. So in essence, the patient or a healthy adult donor can be the actual donor of the cells. And the diagram below, we're showing how a blood cell can be manipulated to revert back to a stem cell-like state, genetically tweaked to be turned into a stem cell, and then coaxed forward to become the desired cell type. In this particular diagram, we're showing that the stem cell is being made into an RPE cell. And this is a diagram that was created by Kapil Bharti at the National Eye Institute, who is doing some really great work using induced pluripotent stem cells to address AMD and potentially other inherited degenerative retinal diseases. So there are other types of stem cells. One type is known as an adult stem cell, and these are found in mature tissue and organs in our bodies, and they're used in our natural repair and replenishment mechanisms. So you skin your knee, you break a bone, and it's stem cells that are able to grow new tissue to repair and again replenish our tissue and our organs. Now, these cells are not so common. They're relatively rare in our bodies, and they have less stemness. That is, their fate is less flexible. They can only become a limited type of cells in our body. There is another cell type that is actually being used in retinal degenerative disease therapy development, and these are called retinal progenitors. And these are stem cells that have almost become the target cell type. So perhaps they've almost become a photoreceptor. And in this case, they're so far down the development path that their fate has been determined. They can't become any other cell type. And they can have advantages in terms of their ability to integrate into a patient's existing tissue, in our case, retina. Now, these cells have less replicability. You can't make billions and billions of cells from progenitors, but again, they do have the advantages in terms of transplantation and integration. So there are different ways that these types of cells can be used to potentially address retinal degenerative diseases. And one of the more common applications is to replace retinal cells that have been lost to retinal disease. So to make new photoreceptors, perhaps rods and cones, or those support cells, the RPE cells, to replace, again, the cells that have been lost. And by replacing these lost cells, the goal is to restore vision. Now, what's coming down the pike are combination therapies where you're replacing both photoreceptors and RPE cells, because often in conditions, especially macular diseases, both photoreceptors and RPE cells are lost, so you need to replace both. Now, another approach that stem cells are used for is called neuroprotection. And this is where we're injecting cells into the eyes of patients, but instead of replacing cells that have been lost, these cells release growth factors or proteins that help keep the patient's existing retina healthy. So the idea is that these proteins can slow or even potentially halt the progression of retinal disease. The idea really isn't to restore vision, though some vision may be restored early on in the treatment, but the idea is really to save vision, to preserve vision. Now, stem cells are also used to model diseases and to screen therapies. You can look at these cells when you make new photoreceptors or RPE cells. You can look at them in a dish. And researchers over the last few years have been making what are called 3D retinal organoids, these rudimentary retinal structures that are really great for modeling disease and also screening therapies. And this circular entity on the right side of the slide, this orange and sort of pinkish lavender structure, is a 3D retinal organoid. And this was developed by David Gamm at the University of Wisconsin-Madison. So another approach that isn't quite as far along in terms of clinical development, but has exciting potential, is coaxing the retina to self-regenerate. Now, in certain animals like amphibians, and when you cut off, or if they lose their arms and legs or their tails, they are able to regenerate these limbs. And it's also true for the retinas. In a human doesn't have the natural capacity to do this. But Tom Ray, a very accomplished developmental biologist at the University of Washington, is investigating ways to bestow this self-regeneration capability in humans. So he's studying this in mice. He's had some success there. And now he's investigating this approach in larger animals. So it would be really great if we could coax the retina to regrow its own photoreceptors. That would remove the barriers of integration and potentially immune rejection that can occur when you're transplanting cells from a different source. So what are the challenges associated with transplanting cells into the retina? First of all, you have to come up with a procedure to get the cells into the fragile retina, and also find a way to orient them properly into the retina so they survive and they integrate. That can be pretty challenging. And researchers like Dr. Gamm and others have come up with these very thin scaffolds. They're biodegradable, which they put the retinal cells on, perhaps photoreceptors and or RPE cells, So they are better oriented in the retina when they're transplanted. And these scaffolds definitely help with survival and integration. And that's what this rectangular structure divided into cubes, it sort of looks like an ice cube tray, is on the right side of the slide. This is a biodegradable scaffold developed by Dr. Gam, again, at the University of Wisconsin-Madison. Now, another thing that we need to be cognizant of is a potential immune response. So the eye is considered an immune privileged structure, but that doesn't mean some type of immune response isn't possible. And that's more likely when you're transplanting allogenic cells, which are cells from another source, from another human. Now, if you're using induced pluripotent cells where the patient is the cell donor, we call those autologous cells, and the immune response is much less likely because the transplanted cells are coming from the patient themselves. Now, to conclude, a couple things I want you to keep in mind. A benefit of most stem cell therapies is that they're designed to work independent of the mutated gene causing the retinal disease. So they are gene agnostic for the most part. What's most important is the existing structure of the patient's retina, how severe their degeneration is. You need to have a reasonably healthy retinal structure still surviving so that cells, when they are transplanted, can integrate and survive. So again, that's the most important thing when considering whether a stem cell therapy will be appropriate for the patient. So finally, to conclude, if you want to learn more about emerging therapies, whether they're stem cell therapies or other therapeutic approaches, you can always go to fightingblindness.org to learn more about these emerging therapies. You can go to clinicaltrials.gov to learn about clinical trials that are underway in the US and overseas. And if you want to get on the radar screen of researchers and companies that are conducting clinical trials, you can go to our patient registry at myretinatracker.org. And that's it. Thanks for joining us.